All right, welcome back to the deep dive. Today, we're going deep on photosystems.
Tiny but mighty.
Yeah, they're these microscopic solar power plants, right?
Exactly. They're how plants, algae, even some bacteria capture light energy to fuel, well, pretty much all life.
And our sources are diving deep into the structure of these photosystems. They're these protein complexes kind of tucked away in specific membranes within the organisms.
And our goal today is to really try and understand how they work, how they actually capture sunlight and transform that into usable energy.
And I think it's fascinating that we're focusing on two specific types, right?
Right, we've got photosystem I, or PSI, and photosystem 2, or PSII. They're the dynamic duo of photosynthesis.
And one thing that just it blew my mind is that, when plants are exposed to both red and red light,
Yeah.
the effect on their photosynthesis is actually synergistic. It's not just like adding the two together. It's more than that.
It's pretty amazing how efficient they are.
Yeah. So let's break down the structure of a photosystem. Like, what are the nuts and bolts here?
Okay, so every photosystem has two main components. You've got the reaction center, which is where the actual photochemistry happens. It's where light energy gets converted into chemical energy.
Okay, so that's the core of the operation.
Exactly. And surrounding that reaction center, you have this antenna complex. It's like a net that captures light energy.
And that's made up of chlorophyll, right? Hundreds of chlorophyll molecules, according to our sources.
Yeah, chlorophyll's a key player. But there are also other pigments in there, all working together to grab light energy and funnel it towards that reaction center.
And it's not random, right? They're arranged in a specific way.
Oh, absolutely. The pigments that absorb higher energy light, they're positioned further away from the reaction center. And then as you get closer to the reaction center, you have pigments that absorb lower energy light.
So it's like a cascade.
Yeah, like a perfectly organized energy relay. This allows for something called resonance transfer, where the energy jumps from one pigment to the next, getting closer and closer to the reaction center.
And if this didn't happen,
well, the energy would just be released as fluorescence, like, just a faint glow. No photosynthesis.
wow. Okay, so let's talk about PSI and PSI. What are their individual roles in this whole process?
All right, so PSII, it's the one that really kicks things off. It mainly absorbs red light, but its big job is splitting water molecules.
Splitting water?
Yeah, it breaks water down into oxygen, protons, and electrons. And this is a big deal. This is where the oxygen we breathe comes from.
Wow.
And the key player in this water-splitting action is, it's called the oxygen evolving complex. It's this cluster of 4 manganese atoms. It's really fascinating.
So PSII, it's like the water breaker, and it supplies the electrons for the rest of the process.
Right. And those electrons, they move on to PSI.
PSI, which absorbs far red light.
Exactly. And PSI's job is to take those electrons from PSII and boost their energy level.
Like a booster rocket.
Yeah, exactly. And then PSI transfers these high-energy electrons to make something called NADPH.
NADPH. And that's important because
NADPH is like an energy carrier. It shuttles energy around the cell to power all sorts of other processes.
okay, so it's like the delivery truck for energy.
Yeah, perfect analogy.
Now, our sources also mentioned something called non-cyclic photophosphorylation. What is that exactly?
Right, so that's the whole pathway we've been describing, the electrons going from water to PSII to PSI, and then ultimately being used to create NADPH.
Okay.
But there's more to it. As those electrons travel between PSII and PSI, they go through this chain of molecules called the electron transport chain.
Like passing the baton in a relay race?
Right, and with each handoff, some energy is released.
Hmm.
And that energy is used to pump protons across a membrane.
Okay.
And this creates a gradient, difference in proton concentration. It's like building up water behind a dam.
And then that potential energy is used
To make ATP, another energy currency of the cell, there's this enzyme, ATP synthase, and it's like a turbine in the dam. The protons flow through it, and that spins the turbine, generating ATP.
Wow. So non-cyclic photophosphorylation, it gives you both NADPH and ATP.
It's incredibly efficient. And there's another cool thing. PSI can actually work independently, too.
Oh.
Yeah, in a process called cyclic photophosphorylation. In this case, the electrons just cycle around PSI,
Yeah.
and it mainly contributes to ATP production.
So both PSII and PSI are essential for oxygenic photosynthesis, the kind that plants and cyanobacteria do.
Right. And it's interesting, Bacteria that don't produce oxygen, they often have just one type of photosystem, similar to either PSI or PSI.
And speaking of types, our sources also mention different types of reaction centers.
Yeah, there are two main types. Type I, like in PSI, uses iron-sulfur proteins to accept the electrons at the end of the chain. And then type 2, like in PSII, it uses quinones as the final electron acceptors.
So different molecules, but both involved in capturing those high-energy electrons.
Exactly, and it's thought that both types actually evolved from a common ancestor.
Now, with all this activity going on, I imagine these photosystems can be vulnerable to damage, especially under intense sunlight.
Oh yeah, too much light can definitely be a problem. It can stress and even damage the photosystems, particularly a protein called D1, which is part of PSII's reaction center.
And that damage can.
It can disrupt the whole photosynthetic process,
Yeah.
and it can even lead to the formation of harmful reactive oxygen species.
So like the plant's version of sunburn.
In a way, yeah. But plants have evolved some clever defenses. They can release excess light as heat, for example, and they also have antioxidant systems to neutralize some of those harmful reactive oxygen species.
Like their own internal sunscreen?
Yeah, exactly. And they can even repair damaged photosystems. For example, if that D1 protein gets damaged, it can be broken down and replaced with a new one.
So it's a constant cycle of damage and repair.
It's a delicate balance.
All right, so to wrap things up, photosystems are these incredibly complex, sophisticated machines that capture sunlight, split water, and generate the energy that fuels life as we know it.
And the fact that different wavelengths of light can work together synergistically, and that PSII and PSI cooperate so effectively, it just highlights how incredibly efficient these systems are.
Absolutely. But it also makes you wonder, with all this delicate balance and repair going on, what might happen if the light environment changes significantly? Like, what are the implications for the survival and productivity of these photosynthetic organisms?
That's something to think about.
Yeah, it's a big question. And that's all the time we have for the deep dive today.
Thanks for listening.
We'll see you next time.